actually talking to you guys about how to use deep neural networks, in particular neural networks that are based on dynamics learning, so neural ODEs and things, and other networks that are related to neural ODEs that can help you learn dynamics in biology. And of course, why do we want to learn dynamics in biology? Of course, biological systems are very dynamic, and I know a lot of us also work on diseases. Diseases are also some kind of dynamic process that evolves. So you can have cells that are in homeostatic states that are just sort of monitoring the tissue, and then they can start to transform into a different phenotypic identity where they can become inflammatory. They can recruit all kinds of other immune cells to come attack the tissue. Or in the case of cancer, they can undergo a transformation where they take on stem-like states and start proliferating. At any rate, there is sort of a dynamic process that unfolds, and that's what causes disease progression, for example. And the question is, can we discover insights at this kind of cellular dynamics level from sort of the data that we currently have, which are largely static snapshots? So this is single-cell RNA sequencing data, maybe the workhorse data type that people use for large-scale cellular discovery. As you may have heard, the Human Cell Atlas has cells of every single tissue present. And there are related technologies to these, whether it's spatial transcriptomics or the multi-ohm kit, which can give you single cell at the same time as ATAC-seq data. These are kind of giving you different views of it. But nevertheless, what they're all doing is taking sort of a static snapshot picture. So it would be like if, I was trying to teach you dance, but I'm only showing you freeze frames of people dancing every 2 minutes or something. Like, you can't really maybe learn the steps. So that's been a key problem in biology that people have understood for a while. And even before deep learning became very popular in biology, there were other methods, like RNA velocity or pseudo-time that would try to do these. But I'm going to try to argue with deep learning, you can take this to the next level. You can actually kind of reanimate and infer real diverse cellular trajectories from static snapshot data to form hypotheses. So single cell data comes with a plethora of challenges, as you know, and we have to keep all of those sort of in mind. So when we're designing these kinds of neural networks that learn dynamics. So I'm just going to go through sort of a regular list that I tend to put up. First off, they're very, very noisy. So one of the solutions that we have to dealing with the noise is to go to a lower dimensional latent space, where if you decode from there, you get rid of the noise dimensions. But you don't have to decode, as I told one of you guys in the last session. Also, cells can't exist everywhere in space. They exist in certain areas where they're viable. So you want to use the geometry of the cellular state space a lot to figure out how things are transitioning. And this is going to be really key. So you want to be able to distill cells down to a state space where you have, where the intrinsic groupings between cells are seen. And so even if you have static samples, you can see there's kind of trajectories happening there. And you can have multiple data sets. You would want sort of ways of integrating those, which I'll talk about a little bit later. And finally, you want to keep all these factors in mind as you go from static data that you either have a single time point of or a few time points, and you want to learn dynamics. So let's get more into that, because that's the key thing that I'm talking about today. So let's suppose that even people who are measuring at an intricate scale, for example, my cancer research colleagues, they typically have just a handful of time samples. They'll have a patient when they're first diagnosed, they might have a patient at surgery, and then they might have a patient at relapse. So there's 3 time points. In mouse models, they sometimes have more, like 4 or 5. But whatever it is, it's very countable on your hands number of samples. And you'll have these point clouds of cells that you've collected at these. So what are exactly we wanting to do? We're trying to say, okay, when we measure these cells, they all die because cells don't survive the RNA sequencing process. So had the cell lived, what would its fate have been? And that would be interesting to us because let's say these are cancer cells and these are, a metastatic type state. And these are a more benign state. You want to understand, where did these metastatic cells come from in the original population? That I've coined as the cell of origin analysis. And that's a very important kind of analysis in biology. And the other thing you might want to tell is, I just couldn't measure all the time points, so can I recapitulate what happened to this whole population? Did some cells start differentiating this way? And then what happened? So this is population level. And this is kind of what we want to do. And there are a lot of things that are going to help us do it. But the basic thing that the neural ODE network is going to do is it's going to take each of these cells and it's going to give the derivative of the cell so that it can be pushed to the next state. So in a way that's sort of optimal and not wasting time by squiggling. And when you push this whole population of cells, which you can consider kind of like a mound of cells, into this next mound of cells, I'm trying to use earth remover's distance type dirt-based terminology here. In the wake of it, you'll leave the single cell transport paths. And that's kind of the overall idea. I'll go a little bit more deeply into it. So just to go into some of the ideas that actually help us here, one of the ideas is the manifold hypothesis that you might have heard of. That even though cells are measured in 20,000 dimensions, they live in a much lower dimensional space, much like this three-dimensional sheet actually lives in 2D if you look at where it is. And that our data is actually sampled from that. So there is a way of roughly learning the shape of this manifold using these samples. If, for example, you created a nearest neighbor graph out of them. And that's kind of the manifold hypothesis. So sometimes it's splayed out with noise, but if you project it back from noise, you still get that. So that allows us at some level to represent cells as graphs or trees. And you can also use methods of embedding graphs or trees into lower dimensional latent spaces in order to have a space where you're following the geometry of the cell. The other kind of concept that I'm going to talk about is a general concept that's useful whenever you have many samples or many time points, and this is a real data set with many time points, is this ability to reason about populations of cells rather than single cells. So when you think about individual data points like this is a fate plot, and each dot here is a data point. So what if we had populations of cells that were captured from all these individuals or at 10 different time points? Could I make a similar plot where the points are actually the cells, or are actually the whole sample? So this idea is lifting whatever analysis you have from the single data point level to the point cloud level. And this kind of analysis is key to both learning dynamics and understanding how the dynamics is different under different conditions, and that's called optimal transport. So those will be some of the key ingredients that are used, but I'll go ahead and tell you how we've built a method for learning trajectories from single cells, and we've had several incarnations of them, basically using this combination of the geometry of data together with the topology of the neural network that we're using in order to learn these. So the pipeline roughly goes as follows. So we have a latent space embedding of the data. In this latent space embedding, we have a network that learns continuous time derivatives. Those are integrated out to form single cell trajectories. We then can take these trajectories, decode them back out into the gene space, and learn networks. And these networks will then hopefully tell us something about perturbations, which we can also capture using optimal transport. So this is our flow for understanding dynamics, and the actual neural network I'll describe today is a neural network that we're still working on improving, but was published in NeurIPS 2022 about a year and a half ago, called Mioflow, or Manifold Interpolating Flows. This is sort of our second kind of very mature deep neural network that does this. So again, the data I'm showing you here is 5 time points from an embryoid body differentiation data set, and we want to learn continuous cellular trajectories. The Mioflow architecture looks like this. First, it embeds cells into a latent space where these graph geodesic distances on the cell-cell graph that represents the cellular manifold are preserved. So that means that if cells are, in a trajectory, they're likely to be close to each other. This sort of thing. The next component is what's called a neural ODE network. And I'll describe to you the base neural ODE network, and if we have time, I'll talk about how sometimes you don't need to do the integration for example, which takes up a lot of time. So let me explain to you what the neural ODE network does, and then I can come back to the schematic to show what it's actually doing. Neural ODE network, basically, instead of, when you give it a data point, instead of predicting something, which is a function, it predicts the derivative of the cell state. Okay, it says how's the cell going to change, which is an instantaneous time derivative, and also you can see it's parameterized by time, so the derivative can change with time. So this is a network that can take in a time point and say how's the network going to change at the very next time point. And because this network gives the derivative, then you integrate it out to give you what would be the cell's actual state at a future point in time. And maybe you've measured cells at that point in time, and you can see if your derivatives led to the right integral and penalize it that way. So just a question, how do we train these neural ODE networks? The neural ODE networks are typically trained by learning the derivative of a dynamic system that when you integrate out, you get the future of a time trace. Because often people have time trace or time varying measurements of a dynamic system, but they don't understand the dynamic behind it. So it's really just trying to learn the ODE inside a dynamic system. So what does it mean to integrate it out? It means you're getting the derivative, but you have to integrate the derivative to get the actual curve. So you have to use an ODE solver to do this. So the ODE solver is kind of inside the neural network end-to-end training. So if you wanted to do it straight across, you would have to backpropagate through all the steps of an ODE. There are clever ideas for avoiding that, like something called adjoint sensitivity, which just estimates the derivative through the ODE solver. So I'm going to skip over that. Just trust that that can be done. But one issue with just, if you just read literature about neural ODEs, you come across neural ODEs being trained on systems where you have all the time traces you need, and you just train it to generate another one as a generative model, usually speaking. But we actually don't have the trajectories of cells. We actually only have these static snapshot measurements. So what can you do? And here's where optimal transport becomes a very key tool. Optimal transport is also familiar to us in one guise as the earth mover's distance. It's like if this was a mound of dirt and this was another one, what is the least amount of work you have to do to push one mound to another? Typically speaking, you have to develop a linear program to solve this. So a transport plan is formulated. The transport plan says, how much of the dirt from each of these bins do I have to spread out to these other bins? And for every amount of dirt moving, I have some penalty. So there's large penalty for moving the dirt far away, and there's small penalty or zero penalty for leaving it where it is. So basically, it's this joint probability distribution, which is called a transport plan, penalized point-wise by this distance matrix. And that's the objective. And the constraints are that the marginals have to equal these two distributions. And you can solve this as a linear program. But if you had many, many conditions, many, many cells, you don't really have bins like we do, then it's very hard to do. Instead, we're actually going to train a neural network to do this optimal transport. And how do we train a neural network to do optimal transport? It turns out there's a formulation of optimal transport that's called dynamic optimal transport, which you can get a neural network to do pretty easily. So what you say is, create me the derivatives to move this cell population to this cell population. And then the neural network might do this. But this is not what you want, because this is not an optimal transport. You're wasting all your time meandering, squiggling. So you just penalize the magnitude of the derivative along these curves. See, there's a penalization of the magnitude of the derivative. Once you put this in, if you hike this penalty high enough, you'll straighten out all these paths, and you can see these are being optimally transported. Not only are they reaching the correct destination, but they're also doing it in the most efficient way. So we've proven that just by adding this regularization to the derivative, you get dynamic optimal transport. And you can do whatever you want to check that the marginals are correct. You can even do Kell divergence. That's kind of weird, right? It's still dynamic optimal transport, no matter how you check the marginals. So we have proofs of this in the paper. Just to give you an idea of that, if you have original point cloud that looks like a Gaussian, you want to transport to an S, a dynamic optimal transport would take these points to exactly where they have to go quickly. They would not slide down the S. And as we train our neural network and we hike up this magnitude of derivative squared regularization, you start getting more and more of them. Okay, so that's the basic gist of what we do. We tell the neural network, push this population so it gets to the next one with as little effort as possible. And as the neural network pushes it, you will get single cell trajectories. But wait, there's another problem. Single cell trajectory has to go along these optimal transport these paths that are plausible. So maybe that would be the optimal transport path in Euclidean space. But it would not be in cellular space because there's no cells there. That means that's not a part of the space that cells exist in. We have to pay attention to the cellular manifold. So we use two tricks for that. The first trick is actually doing the transport in a more intrinsic space of the cells. And we've played with a lot of intrinsic cell spaces. In the Mioflow paper, we talk about a space where distance between cells is the geodesic distance on this cell-cell graph, as measured by a diffusion operator on that graph. More recently, we actually just use our FATE dimensionality reduction method distance. So FATE is a dimensionality reduction method we came from that also turns cells into a graph, but then it embeds cells based on their diffusion probabilities between each other in such a way that the geometry is preserved. I won't go into the details of FATE, you can read about it, but basically we trained this autoencoder to either match the geodesic autoencoder distance or more recently FATE distance so that it's naturally going along the main manifold dimension. So the main manifold dimension here actually looks like this. So its primary direction of motion would be over here anyways. But we have a secondary loss that penalizes going in areas with no cellular density. So that's how we solve the problem of going in a dense area. So now we have this set up again so that a neural ODE is learning the optimal transport paths. We're integrating it out to the next time point where we have observations. And if we don't have time-resolved observations, we just bin them based on pseudotime. And then it transports this, and you can just see if the marginals match. And then it'll give you these single-cell trajectories. Another issue is cell growth and death. So cells aren't just transported. We also want to give it the ability to kill cells or have cells be born. This, too, can go into the framework of optimal transport. You basically have to learn growth or death rates, which it's the same rate. If it's less than 1, it would be death, and if it's more than 1, it would be growth, in order to do what's called unbalanced optimal transport. So once you put these in, this is actually what the transport looks like. As you're going from day 03 to 6/9 to 12/15, you see the whole population is being pushed. And in the wake of the transport, you're getting different trajectories. So this is very important to remember. So this style of method is the only method I know. And there's other methods after we came. I'm guessing we were the first ones to come up with this kind of method with trajectory net. There are other methods out there that do something similar now, but only this style of method gives you single cell trajectories. So things like RNA velocity don't. They're just giving you velocity arrows, and I can show you different differences between that or pseudotime. So what you can do with this is say, okay, these different cells that ended up in different fates, neuronal, endothelial, cardiac, muscle, how did they get there? How is their differentiation path different? So you can look at the dynamic, you can trace these trajectories backwards, just like you can do them forwards, and you can look at how each expression of each gene was quite different in these trajectories. And so then you can tell the gene dynamics that were underlying these cellular dynamics. And so I'm going to skip over this next story just because I want to tell you some other cool thing that we can do with these neural ODEs. But I'll briefly go over it. So one of the systems that I've been studying with this is this mammosphere growth, which is a breast cancer organoid growth. So here there are single cells, you can't tell them. And here they're forming small mammospheres. Here some of them formed quite big mammospheres. But some of these actually died. So you want to know which cells from this day actually really seeded these, right? What was their state? That's the question we're working on studying, and it involves two processes called EMT and MET. And we have this 5-time course we're measuring. And in this study, we applied our earlier iteration of this method called trajectory net. And then we were able to trace back these cells that are highly proliferative at the end. Where do they come from? This is called cell of origin analysis. We're able to refine a signature for the cell of origin, which could be a stem cell signature for this particular kind of cancer. We can also tell how the gene dynamics are different between these two types of end states. But the really exciting thing to us was you can use these gene dynamics to learn networks. The first very simple way we did it was we have continuous gene trajectories that come with these cell trajectories. So we can use a measure called Granger causality directly to learn these gene networks. And this actually worked quite well for us. For this particular data set, we got these gene clusters that seem to go up in cascade, and there's a submodule that crosstalks between these modules that has several known regulators of EMT, and we found a couple of new ones. And so this is listed in our preprint. But one thing as computational people were dissatisfied with was the network we were learning is a static network from these dynamic cells. And so more recently, we decided to use ODEs in another way to learn the dynamics of the gene regulation itself. And this uses what's called a graph ODE. Here we have a graph whose nodes are literally the genes. So this is not in the latent space anymore. You can learn the trajectories in latent space. But then we're trying to learn how the genes actually interact in this latent space. So what do? This graph is input to a graph neural network. And the graph neural network is trained to predict the next value of these genes along a particular cellular trajectory. So it uses a graph ODE, a graph dynamic network, to do that. And how do we know what the very next expression of the gene is? We just use Mioflow. We just make it match Mioflow. So what it's doing as it's doing that is it has a mechanism called attention, which is the same mechanism used in LLMs and transformers. And so when predicting the value of one gene, you look at the values of other genes. And that's how, regardless of what graph, even an uninformative graph we input to the network, we can come out with an idea of the changing regulatory network that's operational in the cell. And we sort of redesigned the attention to be attention not only over neighbors, but also over time, so that you can accumulate some transcripts before you trigger another one. I'm not going to go into the details of this, but basically you have a time-varying attention network that has several parameters. It can support accumulation of some kind of transcript before triggering another one, and it models time lag between neighbors. So once you do that, we have a bunch of systems that we tried it on where we know the ground truth, basically simulators like Nest and Sergio, where we tested it. And some of these comparisons are only done statically because these other methods are static. So the basic way we do it is we look at some kind of averaged graph of ours versus the static graph produced by others, and we measure some kind of gene edit distance. So once you have this, you can reason about perturbation. So if you perturb some node, you can watch that perturbation sort of cascade through the network. And it'll give you time-varying predictions of what should happen after a perturbation. And this might be wrong, but if you actually carried out this experiment, then you can use that as training data to retrain the network to match the results of the perturbation, because you also have perturbed time traces now. The thing that this avoids is testing every perturbation on Earth, because you have this network, and this network has hubs, and those would be the natural perturbations. Hubs in terms of the attention graph of this network. So this is the same graph that we were looking at before, and we tried perturbing genes, and you see you can even do double perturbations, which are hard to do in person. So we've been trying this on various systems, T-cell activations and things like that. The final thing that I just was going to show is, at the end of the day, once we have a bunch of perturbations, for example, in the T-cell system, we got 50 perturbations, you can actually map the space of perturbations like you map the space of cells by also using optimal transport. So optimal transport turns out to be very useful. So for that, we have a graph-based optimal transport based on taking perturbation data from a lot of different perturbations, projecting them onto a graph, and performing a graph-based density estimate. So at the end of the day, our perturbation embeddings look like this, and so we can reason about different classes of perturbations that are likely to have similar results versus different results. So I'm going to just end my talk there. Hopefully I've shown you how to use neural networks to uncover single cell level dynamics, and ongoing progress in also learning gene regulatory networks, because that is actually one of the really good reasons why you might want to learn continuous cell dynamics, is because you actually want to learn the gene regulation and the regulatory dynamics that come out of it. Let me just go to the last slide I have here. The one last thing that I'll say is just the first ODE training part. There's a method in generative modeling that came out that's called flow matching. The idea of flow matching is if you live in a Euclidean space, then you can just draw straight lines in that space and that will be the optimal transport path. Unfortunately, that's not the case for us because we don't live in a Euclidean space. So one of the things we've been doing is there are people who wrote flow matching papers who kind of pretend that it works on cellular data or assume. Maybe if you have like very densely sampled cells. But what we're kind of doing is allowing it to work in a manifold space by modeling the metric on a manifold. So once you have the metric on a manifold, you can draw geodesics on the manifold, and you can do something like flow matching. But that's ongoing work, and we have a couple of publications. This was a workshop publication, and this was a conference publication on a specific kind of metric. So I just wanted to alert you to the idea that I am talking about ideas that come up in generative modeling a lot. But we are using them a little bit differently, and they're not always like portable, black box wise. So yeah, first I talked about neural ODEs, which were proposed for generative modeling, and then flow matching, which doesn't totally work yet, but I think it's getting there.